Welcome to this is the second talk this year, and today we're very happy to have Dr. Liu Yi from Hong Kong University of Science and Technology, Guangzhou. Dr. Liu got his both bachelor degree and master degree from China Agricultural University, and he received his PhD from the University of Hong Kong. After that, he did a few years of a postdoc, and last year he got his current position as assistant professor, also at Hong Kong University of Science and Technology in Guangzhou, China. He has very wide research interests, including isotopic geochemistry, biogeochemistry in coastal groundwater system, seawater intrusion at SGD, numerical modeling on groundwater, surface water interaction, greenhouse emanation from surface water bodies, etc. Today, his topic is submarine groundwater discharge and coastal ocean acidification. Okay, Dr. Liu, time is yours now.
Okay, thank you, Professor Xu, for introducing me. And it's a great honor for me to have this opportunity to share the work I have done in the past years. So let me begin with, so can you see the screen?
Yes, it's working.
Okay. Today my topic is submarine groundwater discharge and coastal ocean acidification. And I'm Liu Yi from the EOAS, the Hong Kong University of Science and Technology, Guangzhou. First, I want to thank my supervisors and my co-authors and also supporting grants and other departments in Hong Kong. Today, I'm going to give talk including an introduction to explain why I conduct this kind of research and my motivations. And then I have give two topics. First one is the carbonate chemistry in the intertidal aquifer, which is the onshore biogeochemistry. And then I gave a topic on the coastal ocean acidification, which is kind of consequences of the groundwater discharge to the ocean. And then I gave a summary. Firstly, I want to emphasize the importance of the submarine groundwater discharge. First, it is a major component of the global hydrological cycles. So you can see the meteorology alters infiltrates into the aquifer and it flows to the sea. And importantly, more importantly, SGD are essential components of the biogeochemical budgets. According to the Nature Review paper by Professor Santos, the nutrients carried by the total SGD is comparable to the river discharge. So it causes a significant influence on the coastal environments. And there's some publication also emphasizes. So as an important pathways and land-ocean interaction, SGD is a bridge connecting the offshore environments and ecology with the onshore activities. So in the coastal ecosystems, so it is not a semi-closed, not a closed system. It contains the onshore processes and also the offshore processes. And it also has a connection with atmosphere processes and open sea processes. A lot of processes that make them connected with each other. For example, SGD and river discharge and sewage and also tidal fluctuation connect the onshore process and also the biogeochemistry in the water columns of the coastal bayment. So the coastal ecosystem is not an enclosed space. Integrated studies considering multiple onshore and offshore processes are of significance to provide a comprehensive understanding of the system. So first, I will give some backgrounds about the seawater pH and carbonate chemistry in the ocean. And the seawater pH is majorly controlled by the carbonate chemistry in the water. So it contains a gas equilibrium between the atmosphere and also the water column, and the two dissociation reactions of the carbonic acid to dissociate with hydrogen ion and also bicarbonate ion. And the second dissociation reaction to produce more hydrogen ion and carbonate ion. And also the carbonate ion will combine it with the calcium ion to have this mineral dissolution and precipitation processes. So this kind of carbonate chemistry controls the seawater pH. So at a different pH, the composition of the dissolved carbonate, carbon dioxide, bicarbonate, and carbonate are different. So there are two very important parameters. One is the total alkalinity, and another one is dissolved inorganic carbon. So the total alkalinity is the measurement of the capacity of the water to neutralize the acid. So it's a combination of all the ions that can receive hydrogen ion. And the dissolved inorganic carbon is the sum of all dissolved inorganic carbon species, including the dissolved carbon dioxide, bicarbonate, and carbonate. So the sensitivity of the seawater pH to the change of the total alkalinity and the DSA is different. From the right figure, we can see that the change of the pH with the total alkalinity is positive, which means the increase of the total alkalinity will raise the pH. And at the seawater pH range from 7.5 to 9, so the sensitivity of the pH to total alkalinity is kind of decreasing here. And for the DEIC, the increase of a DEIC will decline the pH, and it shows the sensitivity is a negative value. So at the seawater pH between 7.5 to 9, and the increase of the pH, the sensitivity to pH to DIC is decreasing. So here, the total alkalinity and DIC can be used as a proxy for the ocean acidification, because total alkalinity can be equals to this much, and the DIC equals to dissolved carbon dioxide plus the bicarbonate and the carbonate. So total alkalinity minus DIC equals to the bicarbonate minus dissolved carbon dioxide. So the bicarbonate in seawater is relatively stable. So pH is mainly determined by the relative change of the bicarbonate and the dissolved carbon dioxide, which apparently can be reflected by the total alkalinity minus DIC. And why we need to use this kind of approximation is because the total alkalinity minus DIC is a linear combination of the two conservative properties, and thus it itself is conservative. So it can be used to do the linear mixing relationships. So here is the global ocean total alkalinity minus DIC relationship with the pH. It shows a very good correlation between each other. So here I give ocean acidification is a kind of has a definition. Ocean acidification is defined as a decline of the ocean pH and calcium carbonate saturation state. So the major causative factor of the open ocean acidification is the uptake of the CO2 from the atmosphere. So we know that since the industrial revitalization, the atmospheric carbon dioxide is increasing and doubled. And so the dissolved carbon dioxide will push the carbonate dissociation to the carbonate and bicarbonate and produce more hydrogen ion in the seawater and cause the decline of the seawater pH. So it has a considerable negative impacts on the global marine organism and substantially modify the ocean biogeochemistry. But for the coastal ocean acidification, there's a lot of land-sourced chemicals input, and it is more complicated than the open ocean acidification. So from the figure, you can see there's a lot of discharge of the waste water from the industrial, domestic, and agricultural activities. And also we have the submarine groundwater discharge. And there are complex biogeochemistry in the coastal ocean, and also it has the interaction between the air and also the water. And it also has exchange with the open ocean. So it has external sources such as river discharge, rainfall, sewage, urban runoff, and also exchange with the atmosphere and also open ocean. It also has interior biogeochemistry such as the photosynthesis and respiration, and also has a precipitation dissolution of the calcium carbonate mineral and other redox reactions. There are a lot of publications in the high impact journals like Nature Geoscience, Nature Climate Change that emphasize the importance of the eutrophication, redox reactions, and other carbonate chemistry. But the SGD is overlooked in most of the coastal ocean acidification studies. So in coastal ocean, seawater pH is highly sensitive to the land-ocean interaction and anthropogenic activities. So as the most important chemical loading pathways for the coastal ocean, SGD has a great potential to influence the carbonate chemistry and ocean acidification. So I'm going to jump to the first topic to tell a story about the onshore biogeochemistry in the intertidal aquifer. So I have conducted a field trip to the harbor of Hong Kong and installed this kind of a permanent sampling and monitoring systems in an intertidal aquifer. It contains a bare sandy beach and also a mangrove soils. And we have this kind of a sampling, and we conduct a lot of sampling. And we have the sampling strategies for the seasonal scale and also the tidal scales, and only in the transition zone. And we measured the pH, DO, ORP, and salinity, and also temperature, and do the titration for the total alkalinity in situ. And then we can calculate the DSC and PCO2 using a CO2 system program. And we also measure the major ions and nutrients for the analysis. First, we need to quantify the SGD flux. And here we use the monitoring system to monitor the water table in the intertidal aquifer, and use the water table difference at the high tide and low tide, and then we can obtain the SGD flux during one tidal cycle. And for the fresh SGD, we calculate the land sea hydraulic gradient and hydraulic conductivity, and then use the Darcy's law to obtain this fresh SGD. And to evaluate the function of the intertidal aquifer, we use this kind of mass balance model in the intertidal aquifer, and we quantify the fresh SGD and the seawater recharge and the SGD, and then we can obtain the production or consumption of the chemicals in the intertidal aquifer. And to identify the pathways of the production or removal of the specific chemicals, we use this kind of non-conservative mixed model in the aquifer. and we assume the seawater has a certain for the chloride has we use it as a conservative mixing in tracer, and if the chemicals is above this conservative mixing, we regard it as a production. If it is below this conservative line, we regard it as the consumption in the intertidal aquifer. So here is some results. First, the groundwater donation and properties for the fresh groundwater here at the land side area, it has a very low salinity and a low pH. It is a little bit oxic to slightly reduced from here, you can see. And for the transition zone between here, between the high salinity zone and the fresh water zone, it is mainly in the mangrove area, and it has a salinity between 1 and 25, and has a pH between 6.5 to 7.5, and it is highly reduced at the surface area with a high organic content in the soil because the mangrove is here. And for the high salinity zone, it's a kind of a bare beach area. So it has a very high salinity over 25, and pH also over 7.5. And it is slightly too highly reduced with the NRP value between -20 to -150 microvolts. As for the carbonates, and here is the PCO2, total alkalinity, and the DIC. And for the freshwater groundwater room, it has a very high PCO2 and a low total alkalinity. And the DIC is kind of combination between them. And in the transition zone, we have a very high total alkalinity and DIC at the near surface area, which indicates the mangrove soil produces the total alkalinity and DIC. And in the high salinity zone, we have very low PCO2 and a very high total alkalinity and DIC at the bottom. For the temporal variation, we have the tidal scale variation and seasonal variation. First, for the tidal variation, we can see from the salinity change, we can see here at high tide, there's seawater infiltration from the top. And we observed the production of the total alkalinity at the top. So which indicates the infiltration of the seawater will promote the production of the total alkalinity and also the DIC. And we also figure out at the high tide, there is a seawater flow of fresh water. From here, you can see from the pH, also from the salinity, you can see the fresh water from this side will flow to the seaside. It is weird because the high tide means there's a high sea level at the seaside. It should be a low hydraulic gradient between the land and sea. But here, the seawater infiltration needs time. So with the sea level change will much faster than the change of the hydrodynamics in the aquifer. So we can observe this kind of phenomenon. As for the seasonal scale variation, we also can see there do not have a significant spatial distribution change of the total alkalinity, but for the mangrove zone at the near surface area of the mangrove zone, it has a difference between different months, which means the production of the total alkalinity in this area is very different. And also we observe the difference at the seaside, which means the mixing between the seawater and also the high alkalinity groundwater is different, which means the hydrodynamics varies between the wet season and dry season. For the DIC, it mainly changes in fresh groundwater room. because the discharge of the groundwater, fresh groundwater will cause a significant change of the DIC, because in this area, the major component of DIC is dissolved carbon dioxide. So we evaluate the inventory and also discharge flux of the carbonate species. We can see from here, the total alkalinity does not change much, no matter in wet season or dry season, but the DIC inventory changes, mainly changes in the CO2 form. And the discharge flux is also has a much higher discharge flux in the wet season, and in dry season is similar to each other. So the major change of the flux is caused by the water flux change. Here we use the mass balance model in the aquifer, and we evaluate the seawater recharge, freshwater, groundwater discharge, and also the discharge of total SGD, and we obtained this kind of production of all kinds of total alkalinity and DIC species in the intertidal aquifer in the dry season and also in the wet season. And we figure out that more than 50% of the total alkalinity and DIC discharged by the SGD is produced in the intertidal aquifer, which means the intertidal aquifer is a great source for the SGD, DIC, and the total alkalinity fluxes. So how can we identify the different pathways of the total alkalinity and DIC production in the intertidal aquifers? We all know that there might be respiration, denitrification, iron reduction, sulfate reduction, and other all kinds of biotic or abiotic processes in the intertidal aquifer, but they have different production to the alkalinity and the DIC ratios. So we can draw this kind of ratio, different processes according to their production ratios of DIC and total alkalinity in this kind of figure. So we can use our data and put them into this kind of ratio. We can compare our data with those processes, and we can see here in the fresh groundwater room, the total alkalinity and the DIC ratio is pretty scattered. So it's not strictly followed to which major process, and it needs more investigation. For the transition rooms, we can say it is between 1 and sulfate reduction and aerobic respiration here, between here and also here, the yellow dots. And also there are high salinity rooms. For the red triangle, it is also between the sulfate reduction and also carbonate dissolution between 1 and 2. So we can speculate that they have this kind of, they may have this major process, but if we want to identify which one is the major or controlling process, we need to do more things. So here we also calculated the production or consumption of the sulfate in the transition room, and compare with the total alkalinity, we found it's a very good correlation between each other, which indicates the maybe sulfate reduction is a major biogeochemical process in the aquifer. But in the high salinity room, in the Bear Beach area here, we don't see this kind of good relationship, and we compare the calcium production or consumption with the total alkalinity. We found a very good linear correlation. So that means the production of the total alkalinity in the high salinity room is highly correlated to the carbonate dissociation. But in the transition zone, it is highly correlated with the sulfate reduction. And there are some difference between the dry and wet seasons. And when we compare the total alkalinity and DIC ratios, and we found that if the total alkalinity and DIC is -1, which decrease the buffer capacity of the seawater. If you can still remember, the total alkalinity is kind of increase the pH of the ocean water, and the DIC is kind of decrease to the seawater pH. If the total alkalinity is minus over DIC is minus than 1, that means the DIC is more than the total alkalinity. That will decrease the seawater pH. So from here we have the total alkalinity over DIC is about 0.8 to 1.1, which is about it's smaller than the nearshore seawater total alkalinity over the DIC. So we conclude that SGD to the harbor will decrease the buffer capacity of the seawater. And when we examine the many studies in other places, and we found the SGD total alkalinity over DIC ratio is smaller than 1, which means that there's a great potential of the SGD to reduce the buffer capacity of the seawater to the atmospheric carbon dioxide. But to what extent SGD can contribute to the coastal ocean acidification? Actually, there is no answer yet. So with these questions, I have come up with the second topic, the coastal ocean acidification in Hong Kong. So I want to evaluate the offshore consequences of the SGD carried DIC and total alkalinity to see what kind of effects can be caused. So I examined the seawater pH in the Hong Kong coastal waters, and there are 76 seawater monitoring stations in Hong Kong's coastal waters, and it has more than 30 years data. So at each point, it is a station. So I extracted the seawater pH data and obtained this kind of trend of the seawater pH, and obtain this map to show the seawater pH decline rate. And the color with the red is much faster than the color with the blue one. And also we found that at the inner area of the bay, it shows a much faster decline rate. For example, DM4 is here, and the MM13 is here. They have the pH decline rate is about 0.0156, which is 2 times faster than the MM13, and it's also much faster than the global value. So that indicates the Hong Kong coastal waters are suffering from a serious ocean acidification processes. So, and also the seawater pH in the Bay Area has a drastic degradation trend. So I speculated that SGD may play a role. So what should I do to identify the reason of the fast ocean acidification in Hong Kong coastal waters? So I draw this kind of conceptual model here, and it includes the river discharge, external water sources such as the river discharge, precipitation, urban runoff, sewage, and also the SGD. Other processes such as exchange with the open ocean here, and also has air, sea, CO2 exchange, and also the interior biogeochemistry. The major one is the photosynthesis and respiration. So to obtain the goal, I conduct several field trips to obtain samples, including the rainwater sample, seawater sample in dry season and wet season, and also the river water samples, and the urban runoff, and the stormwater sample, and the beach groundwater, and also the sewage. And also collected data from the literature and also from the government department. I collected fresh groundwater data points about 500, and also marine quality data from the EPD seawater monitoring stations, and also the river water discharge and quality data from the EPD, and also the meteorology data from the Hong Kong Observatory, and other data such as the tidal level from the Hong Kong Observatory, and also the atmospheric carbon dioxide concentration in Hong Kong. And here to evaluate the fresh SGD, I use the water budget analysis in different catchment. Here is a kind of sources and sinks of the water budget analysis. The source is the leakage from the water mains, and also the rainfall infiltration. The sinks including the evapotranspiration, catch water. Hong Kong also collected the rainwater from the catchment for other purpose of use, and also river discharge, and also the cutoff by the impermeable urban areas. We named the urban runoff, and to obtain the fresh SGD. Here is a kind of how we got the precipitation, catch water distribution, and also the leakage from the water mains, and also the river discharge. Here is the river network and the evapotranspiration from the MODIS, and also the urban runoff extracted from the map. Finally, we obtain the external water fluxes, and here you can see at each harbor we have different combinations. So you can see at the majority of the harbor, we obtain the saline SGD, the major water source. And also the rainfall water is also important, not some bay segments. And here we have the sewage, the most important water source, because majority of the water treatment plant in Hong Kong discharge here. So it has the majority of the water discharged in this area, in 6 areas. After that, we compare the SGD with the volume of the harbor, and have this kind of a relationship with the decline rate of the pH in the harbor. And we obtain this kind of a very good linear relationship, especially in the semi-closed space, which indicates there's a great potential for the SGD to contribute to the pH decline, but it does not mean it is true, because the correlation does not mean anything. So we want to quantify the pH decline rate induced by the input of different water sources. Here we record the total alkalinity minus DSC as a proxy for ocean acidification. We have a very good advantage for use this proxy, because we can do the linear mixing. We use the external water sources and also the bay water to do the linear mixing, and then we can evaluate the pH change induced by the external input of the water, like SGD river and also other water sources. So here we obtain our own relationship between the seawater pH at 25 degrees with the total alkalinity minus DIC. And you can see no matter in dry season or wet season, it obeys this kind of a linear relationship. So here we obtain the different waters and their representative total alkalinity minus DIC concentrations here, like rainwater, urban runoff, fresh groundwater, saline and groundwater, and also river water. And we can see the total alkalinity minus DIC in groundwater is much lower than other water end members. And the SGD contributes the most negative total alkalinity minus DIC flux to coastal waters. And we use this equation to obtain the total alkalinity minus DIC flux to each harbor, and we can see the SGD, the yellow one, contributes the most nearly in all coastal water segments, except here the sewage contributed the most. And if we want to evaluate what kind of effects the external water can cause, we must within the residence time of the water in the harbor. So we use a tidal prism model, which is usually used in the estuary area, and it is evaluated the exchange during each tidal cycle. And for here we have some segments with two outlets, and then we need to add the tidal prism plus the outflowing of the during the flooding tide. Here, fortunately, we have this kind of a stream prediction system to predict the seawater circulation in Hong Kong coastal area, so we can obtain this outflow during the flooding tide. And another thing is the stratification during summer and mixing in the winter. So here we add a mixing coefficient in this term, and also the return flow factor that the water flow out and will get back at the next tidal cycle will be evaluated according to previous study in Hong Kong. And this is very important. And to evaluate our calculation, and we compared our study with the previous study in Hong Kong coastal areas, and it is a fresh SGD is comparable to each other, and total SGD also within the range. With other studies using different methods like the 300 member or Darcy slow or the radon or the seepage meter. And we also compare the residence time calculated use a modified tidal prism model with other methods such as the numerical model and also the radium isotopes, and we go obtain these comparable results, and that indicates the accuracy of our estimation in this study. And use this kind of linear relationship with the pH. We convert the pH decline in seawater induced by the different external waters within the residence time. We can see the SGD, the green color dominates in all of the coastal water segments except the south, where the sewage discharge is more important. And besides the external water input, I also evaluated the air-sea CO2 exchange. I use this kind of model, which is dependent to the solubility of the CO2, which is a function of the temperature and salinity, and also the gas transfer across the sea and air interface, and also the gradient of the PCO2 in water and also in the air. And here is the results. So we can figure out that at the western side, the CO2 is outgassing, which cause, and in the western side, the CO2 is kind of absorbing into the seawater, cause negative effects to the seawater pH. As of photosynthesis and respiration, which is a major process, biogeochemical process in the water column that affects the seawater pH. Here, we use the AOU, apparent oxygen utilization, and with time, and to calculate the production and the consumption of the DIC and the total alkalinity, and through the difference between the production and the consumption of DIC and the total alkalinity, we can obtain this kind of figure to obtain the total alkalinity minus DIC flux, and then and we can convert this to the seawater pH change. And here is the total alkalinity minus DIC change in seawater at the different coastal water segments. And we can see at the east side of Hong Kong coastal waters, it is in the True Harbor, the photosynthesis over the aerobic respiration, and others kind of respiration is dominant. But the respiration in the western side is much stronger than the eastern side. And for the Bay Open Ocean Exchange, which is the mixing loss of the pH, and here we first convert the pH to the total alkalinity minus DIC, and we use this kind of mixing loss equation to calculate the loss of the total alkalinity minus DIC during the residence time. So here is the mixing loss results, and here is the total alkalinity minus DIC a flux at each day, and we can see at the eastern side, it is losing the total alkalinity minus DIC at each harbor, and at the western side, it's kind of gaining the total alkalinity minus DIC, which means it's gaining the pH during each mixing. But on this side is kind of losing, it's decreasing pH during each mixing. So when we put all the processes together and to compare the pH induced by the change of different processes such as the SGD and air-sea CO2 exchange and aerobic respiration and photosynthesis, and also the mixing with the open ocean, we can evaluate the relative contribution of each processes. And you can see in the harbor area like the Tulu Harbor, Mears Bay, Port Shelter, and also the Deep Bay, the SGD contributes ranges from the 8% to the 47%. But in the other places with two outlets, and the SGD contribution of the SGD is less than 5%. So that means SGD is maybe only important in the semi-closed bay area. And is that true? Really true? But when we compare the neutron flux carried by the SGD, because the aerobic respiration, from here we can see that our aerobic respiration is most important in majority of the coastal waters. So, and the aerobic respiration is closely related to eutrophication by previous studies. So when we evaluate the neutron flux carried by SGD, we figure out that in majority of the harbors, the SGD contributes the most nitrogen and also the phosphorus to the coastal waters, except where the sewage discharges the most, like the 8 and 6 and also 4, where the Hong Kong sewage disposal area. So we may speculate that SGD also play a significant role in the photosynthesis and respiration process. So here's my summary for the two studies. The one is SGD contribute directly to the ocean acidification by discharging more DIC than total alkalinity in the semi-closed bay area, and SGD may also contribute indirectly to the coastal ocean acidification by strengthening aerobic respiration through the discharge of the neutron fluxes. And thank you. This is my end of my talk. And here I have an advertisement for my department. We are hiring PhD students in all kinds of areas. And if someone has interest in, please contact us. So, Professor Xu, this is my talk. Thank you very much.
Very great talk, Dr. Liu. I bet everybody wanted to ask you questions. So now everybody, if you wanna ask questions to Liu Yi, just unmute yourself and ask. Isaac.
Hello, thanks for the great
Hi.
for the great presentation. Looks like you have lots of data and lots of interesting questions and answers. So my question is about that indirect effect. You're saying that SGD is stimulating respiration, right? Therefore, that should be dropping the pH. That was your second conclusion.
Yes.
So I think my question is like, if you throw nitrogen in the mix, that's going to stimulate photosynthesis, therefore remove CO2 and increase the pH. So yeah, do you have an idea about that? How that may work if you throw nitrogen or phosphorus in the mix?
Actually, the input of the nutrients actually strengthen the photosynthesis, but at the same time, it provides more food for the respiration of the like the algae or the microbes. So from previous study, and it's not from my study, from the Professor Chai Weijun, they indicate the eutrophication is much more important than like the CO2 exchange with the air. So this conclusion is mainly according to their finding, and I do not do too much investigation on this part, but I think that's the kind of thing I need to do in the near future.
Yeah. Thanks, Ian. Great talk.
Thank you.
Okay, Emma.
Hi, thanks for a wonderful talk. I had a question that might be a little basic, but in my PhD right now, I'm also trying to estimate residence times using a tidal prism model. And I noticed in your equation, you have freshwater flow as a separate term. And I was wondering, I've never seen that before, and I was wondering if you could explain that equation a little bit.
You mean the groundwater flow in the term?
So in the tidal prism residence time, you have in the denominator, you have the I term.
Yes.
And I've never seen that term before, and I was wondering if you could explain why you had it and what exactly it is.
You mean here, this one?
Yes.
Actually, it is not very important when you compare the magnitude of the inflow of the fresh water with tidal prism. It is not very important. You can remove it.
Okay, but if you had a system that had large freshwater inflows, would you want to include it in the denominator?
Yes, that depends on the comparative proportion of the fresh water input and also the tidal prism.
Okay, cool. Thank you.
Okay. Thank you.
Okay, anybody else? Natasha, go ahead.
Hi guys, great talk. I really enjoyed it. So I have a question regarding to the age of this carbon that is recycled through the system. In other words, if you're saying that SGD promotes photosynthesis, that means that the system is dominated by very young in age carbon. Is that correct statement? Is it fair statement?
Here I just use like a steady state comparison, so I don't have like that kind of a time.
I'm not suggesting to include time, but what I'm suggesting is to look at the ages of the carbon that is recycled through the system, which we might answer Isaac's question of if you put more nutrients, is that going to be, you know, how the system is going to be affected, because it's a matter of balance of old carbon that is stored in the shallow aquifer versus newly produced through photosynthesis. You know what I mean?
You mean
So
the carbon?
how deep is your aquifer system?
You mean the first topic?
Yeah, on the first topic, I don't remember, you show those contour plots where you show the variations of total alkalinity and other parameters. And I don't remember how deep they go below surface.
Let me share the screen first.
Yes, thank you.
You mean this one?
Yes, how, on the vertical scale, you know, how
Vertical scale is about 1 meter.
So it cannot be all carbon deposited there.
Yes, so when you compare this one with
Okay.
this one, you can
Mhm.
see it's only about 3 hours or 5 hours, and the increase of the total alkalinity is obvious.
Yeah. Okay. Yeah, that answers my question, because if you know the system is tapping into some old carbon, it would have been deposited in deeper sediment layers. But in
Yes.
this system, what you're looking at is just the shallow aquifer, which
Yes.
recycles the modern carbon. And therefore, probably in some seasonal situations, you would have net zero of carbon export, and some yeah, it will vary because of because of nutrient inputs. Yeah. Thank you.
Okay. Okay, thank you for your questions.
Okay, anybody else? Gloria, you wanna ask a question? I saw a small hand in front of your name. No? Okay, so Liu Yi, I have a question.
Okay.
When I kind of remember which story it is, but in one of your slides, you're showing the reason, what is the cause for the production of DIC in the aquifer, and you're showing the arrows, different factors, the arrows pointing to different directions, and some arrows are longer, some arrows are smaller. So my question is, where? Yes. So how did you decide the errors?
Oh, actually, the length of the arrow is does not have any meaning. It's just like the ratio between the total alkalinity and the DIC matters.
But it has actually the meaning like denitrification, calcium carbonate dissolution. It actually have something. So something, right?
That's only represents if it's longer here. That represents the stronger intensity of the production.
Okay, but how about the direction? Why is going to this direction is meaning to the denitrification or sulfate reduction?
So here you can see from this equation you obtain and this equation. So at each reaction, you maybe we assume one more of the organic matter that will produce the total alkalinity and also the DIC, and they have this kind of ratio about 0.9.
Okay, so the direction is very precisely.
Yes.
It's the exact direction. Okay. Okay.
Yes, it's according to the equation here.
Okay. Thank you.
Okay.
All right. I didn't see any more questions. So, right. You guys don't have any more questions, right?
Okay.
Okay, so if you don't have any more questions, I'd like to thank you again. The very great work.
Okay, thank you.
Okay,
Thank you,
so
everyone.
bye-bye.
Bye-bye. Bye-bye.